All right, let's get going. So, today we are looking at the innate and adaptive immune system. This is lecture 1.2. I will say this is not all of lecture 1.2. We have the extravasation video that's also included in this, so give that a watch as well if you want that extra exposure to the material. So yeah, we're talking about the innate immune system and the adaptive immune system. We're going to define those terms, and through the defining of those terms, we're going to learn about the cells that carry out the function of these two systems. All right, so let's get to it. The innate system is what we're starting with. This is your immediate and non-specific response against any pathogens. Okay, so anytime something foreign or damaging enters the body space. All right, so again, this is the quick, immediate response. However, with that rapidity, with that quick response time, we sacrifice specificity. Okay, so it doesn't really these cells aren't going to know what disease that we're fighting. They're not going to have any sort of, context other than damage and inflammation. That's what they're dealing with. So basically, all they're responding to is "ow, ow, come help, ow." That's all they're really hearing. They're not, they're not getting like a suspect is 5'5" with a mullet and a tattoo on his forehead. Like they're not getting specific details of the threat. They're just getting an owie, come help. That's all they're getting. So how do these cells react, or what are the cells in the innate system going to be doing? Well, let's talk about each of them. All right. So you have your neutrophils. That is your first player here. That's my drawing if you like that, but the representation of the cell doesn't do you much good. It's more so all the attributes. They are commonly called first responders or EMTs because of how quick they get to the site of inflammation or injury. They're going to destroy pathogens via phagocytosis. Again, phagocytosis. So phago means eat, and cytosis means like having to do with a cell. So this is just a cell. This right here is a cell eating, in this case, a bacterium. So a lot of your innate cells have this capacity to engulf, foreign entities, engulf foreign debris, and deal with it as they see fit. They also release antimicrobial peptides and ROS, reactive oxygen species. First, let's talk about these antimicrobial peptides. These are going to come up a lot in this course. We're fighting microbes in immunology, so we're going to call those AMPs, antimicrobial peptides. So get used to me using AMPs because it just is going to come up a lot, and this is one of the acronyms. I hate to use them, but it's one of the ones that I'm gonna ask that you get comfy with. Less so reactive oxygen species. These are certainly important, but they delve a bit too into the biochemistry realm, and I wanted to make this class more digestible and not focus on that kind of stuff. But just for like a 30,000-foot view, if you have oxygen anions floating around in any type of metabolic system, these are going to really wreak havoc in any type of cellular machinery, including our own. So they're really good at fighting bacteria, but if we have too many reactive oxygen species around, it really damages us as well. So yeah, too many neutrophils is actually a bad thing. Neutrophils also have a cool special ability. They can catch pathogens in a net, a neutrophil extracellular trap. Now, this special ability is sacrificial in nature. It leads to apoptosis. So, the creation of this net, the the net they make is composed of their genetic material, their DNA. So you can't really live without your DNA. So these cells die in their attempt to try and catch these pathogens and slow them down. We thank our neutrophils for their sacrifice. It's very nice of them. They are the most abundant white blood cell in circulation. If you take a sample of your blood, you are going to find some neutrophils. Some of these other cells we talk about are very, very rare, so this is definitely your most abundant. They're very short-lived, as you could imagine, with that apoptotic behavior and just their first responding nature. There's high turnover, but very short lifespan for these guys. And then lastly, a cool fun fact, or a little morbid fact if we're personifying them like I am. Since so many of them lay their life down whenever there is any sort of inflammation or damage, pus is just dead neutrophils. So if you've cut your knee open and you just had that white filmy stuff underneath the scab, that's thousands and well, more so millions probably of these neutrophils that have laid their life down to try and protect you. So again, thank you so much. Thank you so much for your sacrifice. We love you. We love you. Oh, man, I tried to do the I love you. There we go. All right. Next up is the macrophage. Okay? So, yes, our neutrophils did a little bit of phagocytosis, apart from their other cool abilities. But your macrophage, macro means big, phage means eater. So their main role is phagocytosis. Okay? They hang out in the tissue space, and they hang out for a while, cleaning up microbes and debris. So very much janitors, wound cleaning and maintenance. Another really cool aspect of these guys is antigen presenting. So these are one of your antigen presenting cells. I want you to hold that in the back of your head for the remainder of this video. APCs, antigen presenting cells, are really, really important in terms of recruiting the more specialized cells. We need these cells to go and tell the hospital, what surgeon we need, or what specialist we need. So the antigen presenting cells are going to communicate that, and we'll talk about that more here. But our macrophages are one of them. We'll talk about that more with the dendritic cell, which is up next. So, very similar to the macrophages, your dendritic cells sample your surroundings via phagocytosis. So again, just eating the debris and microbes they come across. And after encounter with the pathogen, they'll present their findings to the adaptive cells. Now, we haven't talked about them yet, but our adaptive cells are the B and T cells. Okay, we'll talk about those in like 5 minutes, though. So just hold those. Get that out of the way. Okay, so what they're going to do after these dendritic cells have eaten a microbe or a pathogen or whatever, they are going to take pieces of it and present it on their arms and say, "Ooh, look what I've got." And dendritic cells have another ability. They have a very high migratory capacity. They can travel throughout the entire body. So let's say you get a little boo-boo, I don't know, let's say in your knee, a dendritic cell here will travel to the lymph nodes. You have lymph nodes in like every vertex of the body, so let's just say it makes its way to your hip. Okay, it'll get to the lymph nodes there, or whatever closest lymph node is, and whatever example that I could use. And that dendritic cell will go to the B and T cells and say, "Hey, do you know what this is? Do you know what I have on my surface? And do you know how to deal with it properly? Because I'm just innate. I don't have the credentials, but a B or T cell does. So let me put that in non-transient ink. There we go, that's better. So that dendritic cell is going to go to the lymph nodes and present its findings. And hopefully something happens after it presents those findings. So it's the big bridge from the innate system to the adaptive system. Remember that dendritic cell has no idea what it's found, but it knows that somebody else might know what it's found, if that makes sense. So it's going to the pros. Say, hey, pros, do you know what I find? Or do you know what I've found? All right. A couple more innate cells, 3 more innate cells, and then we'll go to adaptive. So these are a little weird. So your NK cells are up next. These are technically from the adaptive side of things, but I want you to forget that. We'll talk about that later. They're absolutely part of the innate system in terms of behavior. Their lineage is a little confusing, but they are innate cells. And what they do is they can detect if something is virally infected or if it is transformed. That's the fancy bio way of saying tumorogenic. So if it's a tumor cell or a virally infected cell, they can detect that and they release these nasty compounds, perforins or granzymes, and induce cell death. The way they do this is recognizing that the cells at hand do not have a self marker. And we're going to talk about that idea of a self marker in a couple units. But all of your cells have a specific signature about them. And whenever they're virally infected or whenever they have a tumor appearance to them, well, that changes what their signature is. And NK cells are really good at sleuthing that out. Let me draw a little magnifying glass. That's terrible. Whatever, you know what I'm trying to do. Yeah, so they're detectives. Sure. So they can determine if something has lost that self-determination marker. All right. Then we have your mast cells and your basophils. These are 2 different types of cells, but I'm just going to lump them in together. They really have a very similar effector profile. They behave very similarly. They both contain granules like histamine and heparin. And these 2 compounds, I'm sure you've heard of antihistamines before. So these guys are really annoying in the allergy and especially the anaphylaxis field of things. Because histamine and heparin, what they do is they cause vasodilation. So let me get rid of this. Let me try this drawing again. So vasodilation, you have a blood vessel. Now it's much bigger. So the problem with that is a bigger vessel, that's going to drop blood pressure and just increase flow. And if it's to somewhere that we don't need, or sending fluid to somewhere we don't need, then that can increase swelling, congestion, that kind of stuff. It's also going to increase vascular permeability. So obviously, if this is a blood vessel, we have blood shooting through. But sometimes we need the vessel to be porous, especially if these blood cells need to go fight something. But in cases of allergy and anaphylaxis, if there's too much histamine, then I'm going to get really, really leaky vessels. And I'll start to lose fluid to the tissue space. So that's just a quick blurb into how these guys can be a little annoying. Now, they are really helpful in containing responses and recruiting white blood cells when necessary. But these are definitely the principal players in allergic reactions. All right. And the last immune cell, or innate immune cell I'm going to talk about, are the eosinophils. These are extremely effective against multicellular parasites. Okay? So all the stuff we've been talking about thus far have been little, little amoebas, little bacteria, little strings of nonsense, unicellular nonsense. If I am also just a neutrophil, let's say I'm a neutrophil right here, and I'm secreting stuff onto this, well, we're at a 1-to-1 scale. I have just as much matter as the bacterium that I'm trying to fight. A worm, like if you want to think like a tapeworm or a worm that's just on the sidewalk, yes, those are small relatively, but at the cellular size, these worms would absolutely dwarf, a little cell trying to fight it. So we have specialized cells called eosinophils that have their own specific proteins that are meant to damage multicellular parasites. So there you go. Granules contain toxic proteins that are just for threats that are too big to be phagocytized, like these worms here. Now, a cool thing, and we're going to talk about this, I think, in two lectures. These also contribute to allergies, not as much as those mast cells and basophils. Those are your typical allergies, but absolutely eosinophils do. And the theory is that they're bored. I don't know about you guys, but I find worm infections to be pretty rare. Like, sure, you got like pinworms and stuff like that, but in terms of the modern hygienic world, we don't really see worm infections that much. So there's a theory that these guys are bored, and because of that, they're hyper reactive and contribute to some allergies. So we'll talk about that a little bit later, though. So yeah, whenever I say eosinophils, I want you to think about worms. All right. Now we're into the adaptive side of things. This is the last slide. So your adaptive immune system, it needs time to catch up. It needs a chance for one of those antigen-presenting cells to find the threat and come present it to the B or T cell. So because of that, it is delayed in nature. But once it is activated, it is powerful and very specific. So let's talk about the cells that do this for us. And we'll talk about antigen presentation a little bit more as we go along. So don't stress too much about that concept. All I want you to know is that is how we link innate immunity to adaptive immunity. So your B cells are your first branch of your adaptive immune system. They originate and mature in the marrow. All of your blood cells originate in the bone marrow, but your B cells mature here, and they're going to migrate through the blood and lymph as naive B cells. So they're mature, but they're naive because they haven't found the exact pathogen that they're fighting. And I guess now is as good a time as any to note that B cell, every single B cell is going to fight one antigen. It has a 1-to-1 relationship with a bad guy. So a B cell only has one target that it can ever attack. Okay? That's how specific we're at. Just one string of amino acids or carbohydrates. It has one signature that it is going after. One antigen. Until it finds that one antigen, we're going to call it naive because it hasn't found its soulmate or its soul enemy. So they recognize antigens presented by those innate immune cells, those APCs. Once it is activated, once a B cell is activated, two things happen. Clonal expansion. Clonal expansions. That's exactly what it sounds like. I make an expanse of clones. I just clone the absolute heck out of this cell. Let's think about it. If that B cell that's made to fight, one thing and one thing only, here's my little B cell, and it met its love, or its sole enemy, whatever bacteria, whatever virus it wants to fight. Well, now my body knows that the soulmate connection has happened. So I just make a billion. Let me just do some scientific notation instead. I don't know, something like that. Once that activation has occurred, I know I need millions and millions and millions of that B cell. So that's clonal expansion. Activation occurred, now I need to make billions of you. Now that I have billions of you, I need to do what B cells do. And what B cells do is they make antibodies. They make antibodies. So that's these little things I have hanging off this cell here. We'll talk about these a lot more next unit. All you need to know right now is that when a B cell is activated, it makes antibodies against whatever target it's bred to attack. So how this looks like, let's say this B cell was meant to attack this specific bacteria. We'll just call him Jim. Okay. It's not going to attack anybody else. It's not going to attack Jerry. It's not going to attack Jack. It's only going to attack Jim. Okay? And how this works is the B cell is going to shed these receptors. And now those antibodies, what they do is they just tag onto Jim. And when something is tagged, it makes it way easier to clear. Your innate system no longer has to guess, no longer has to just survey. Anytime it sees an antibody hanging off something, it's like chocolate glaze. We know it needs to go. It tastes better now. Just get it out. So that's what antibodies do for us. And we'll talk a lot more about them later. And a cool thing, your B cells can actually perpetuate this response. And even if they find things before your innate cells do, they can act as antigen-presenting cells. Okay? So that's pretty cool. So to tie everything back, your antigen-presenting cells, that's your macrophage, your dendritic cell, and your B cell. So those are your three potential APCs. All right. So that's your B cell, and now we're going to finish off with the T cells. We talk a lot about these in Unit 3. Unit 3 is the big T cell unit, but I definitely want you guys to understand them and set the foundation now. So they originate in the marrow, and then they mature in the thymus. Now I mentioned the thymus day one. It's that organ kind of in front of your trachea and your heart that starts to involute by puberty. Involution just means kind of goes away and starts getting replaced with fatty tissue. We recognize antigens presented by those innate cells, just like those B cells. But with T cells, we have some cool subclasses, if you will. Your helper T cell is the first one we'll talk about. And then when I say helper T cell, I want you to think of like a conductor. Somebody with the baton on the stage. These really, what am I trying to say? They secrete a bunch of signals and a bunch of chemicals that orchestrate the immune response. If we don't have these, then B cells don't get activated properly. Other white blood cells aren't recruited as well. That's another big thing. We get more blood cells in the periphery here. So your helper T cells are the generals. They make sure everything is occurring in the correct way and that all the players that are needed are there when it's necessary. Your cytotoxic T cells, I like to think of them as assassins. or hand-to-hand combat. Okay. With these, they're going to go right up to the infected or abnormal cells and get rid of them. With all these other cells, that's why I was pausing. I was trying to figure out how to say this. With all the other cells, there's, you're secreting signals, or with the B cells, they were sending antibodies out. With cytotoxic T cells, the cells go right up to these cells and get rid of them. So, I don't know. I like to think of them as just cells like, I don't know, cells holding a knife or something, or a sword. They get rid of them directly, and they use the same kind of compounds that the NK cell does, those perforins and granzymes. I'll go ahead and write those down again. And they get rid of whatever has been infected. These are really, really good. Just like the NK cells, they're crucial for viral infection and cancer defense. In fact, in a lot of cancers, or not in a lot of cancers, but one of the experimental treatments that's going on to treat cancers is they take populations of your cytotoxic T cells out of your body, and they just breed them up, and then they re-inject them back into the body to help fight whatever cancer that's being fought. So those are really, really cool. But I'm going to say I think these steal the show. This is the last cell I'm going to talk about, your regulatory T cells. These suppress overactive immune cells, and this is very, very important because this prevents autoimmunity. And there's a lot of reasons why we don't want these cells just, running unregulated. As I mentioned with neutrophils, and as I just mentioned with cytotoxic T cells, whenever they're activated, they have to secrete a lot of dangerous compounds. So we want to make sure that is kept in check, and our regulatory T cells do that. They make sure that the cells are not overreacting too much. Okay? So those are the cells that we're going to talk about, and then we have the extravasation component of this day as well. But that's all I wanted to talk about today. All right. So thank you guys very much for listening.